mRNA processing. After transcription of eukaryotic DNA, the transcript is known as pre-mRNA. Enzymes in the nucleus modify pre-mRNA before the genetic messages are sent to the cytoplasm. This is known as mRNA processing. During mRNA processing, both ends of the pre-mRNA are altered. Some interior sequences of pre-mRNA may be cut out and other parts spliced together. Alteration of mRNA ends. The 5' end of the pre-mRNA receives a molecule known as a 5' cap or GTP cap. The GTP cap is a modified guanine molecule, 7-methylguanosine, highlighted in red here. It is connected to the ribose sugar of the last nucleotide here via a triphosphate bridge. The 3' end of the pre-mRNA gets a poly-A tail. This tail is a series of adenosine A nucleotides, which you can see here. So here's the original pre-mRNA. Here it is with its GTP cap on the 5' end and the 3' tail added. The modifications to the ends of the pre-mRNA have several functions. Facilitate the export of mRNA from the nucleus to the cytoplasm. Protect mRNA from hydrolytic enzymes, which are catalysts for hydrolysis reactions that break bonds using water once it is in the cytoplasm. And help ribosomes attach to the mRNA so they can be translated into a protein. mRNA splicing. Most eukaryotic genes and their RNA transcripts have long, non-coding stretches of nucleotides that lie between coding regions. These non-coding regions are called intervening sequences or introns. The other regions are called exons because they are eventually expressed and are usually translated into amino acid sequences. mRNA splicing, also called RNA splicing, removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. So you can see the primary transcript with the 3' poly-A tail and 5' GTP cap with both exons and introns. The introns are excised, removed, and the exons are spliced together to form this mRNA molecule. This is an example of a pre-mRNA becoming a final transcript. Here's our DNA genome that's transcribed into our primary RNA transcript. Then we add the GTP cap, the 5' GTP cap, and the 3' poly-A tail. Then we remove the removal of 5 introns and then splice together. And then at this point, we still have 2 additional introns. So the removal of 2 introns spliced together. And this is our mature mRNA, which is then exported to the cytoplasm. So you see here that these introns are removed in 2 steps. Alternative RNA splicing. Eukaryotic genes can code for more than one kind of polypeptide protein, depending on which segments are treated as exons during RNA splicing. Prokaryotes have no introns and no alternative splicing, so they follow the one gene, one polypeptide rule. This is a major difference between prokaryotes and eukaryotes. Due to alternative transcription sites and alternative splicing in eukaryotes, one gene can give rise to several polypeptides, so a human's 20,000 genes could potentially encode 100,000 polypeptides. Alternative splicing allows the number of different proteins an organism can produce to be much greater than its number of genes. So here's an example. Here's the original DNA sequence. Here's the pre-mRNA transcript with the GTP cap, adenosine tail. And you can see we have color-coded some sequences where some can be introns and others can be exons. So look at how we've spliced together the mRNA on the left. We've kept everything in orange, UUU, AAA, UUU, AAA, which codes for the amino acids phenylalanine, lysine, phenylalanine, lysine. That's the resulting protein. Or if we alternatively splice what you see in green, GGC, CCG, GGC, we get lysine, proline, glycine, which is different. These are two different proteins. Alternate splicing can dramatically change the length and or the sequence of the polypeptide chain that will be made. Entrance into the cytoplasm. After the finalized mRNA transcript is complete and correct, the pores in the nuclear envelope allow it to pass to the cytoplasm, where it can be translated into proteins by ribosomes. The nuclear pore is a protein structure that controls the traffic flow of the nucleus. Each nuclear pore is made up of hundreds of individual proteins that ensure only mRNAs with proper caps and tails can make it to the cytoplasm.